Welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I'm hosting this podcast from America's Heartland in St. Louis, Missouri. On June 15th, 2022, we talked with Grant Butschek, a graduate student at Johns Hopkins University in Baltimore. He received his bachelor's in biology from the University of Georgia. He is currently examining how temperature differences in the microenvironment of the respiratory tract affect function of the mitochondria and virus infection.
Thanks for speaking with us today. Can you tell us a little bit about yourself?
Sure. I'm Grant Butschek. I am finishing up the third year of my PhD in Dr. Andy Pekosz's lab at Johns Hopkins University. Yeah, I'm originally from Georgia. I did my undergrad at the University of Georgia and did a year as a technician after graduating. And then following that year, I moved up to Baltimore, joined the program I'm a part of now, and got started on my PhD research.
Great. And can you tell us how you first became interested in science and then virology?
Yeah, I don't have one of those kind of storybook tales as far as how I got into science to begin with. It's just always been something that I've been drawn to. And it's always kind of somebody who I think through that course of the undergrad years where you're deciding your career and everything, I was always worried that the career that I chose would be eventually would kind of let me down. I'd get bored of it or something like that. That was my biggest fear. And science was definitely the area that always kind of helped calm those nerves. It's you can definitely spend multiple lifetimes within a single field of research and have just so much to explore. So I think that was something I've always been drawn to and always made me feel that there was always going to be enough there to entertain me. And now I'm finding it does a whole lot of other emotions as well, but no. So that's what drawn me to it to begin with.
And then how did you sort of get to your specific institution and your specific PhD lab? What drew you to those particular places?
Yeah, so I had a little bit of a winding road, I guess you could say. So I started out my undergrad research at UGA was focused in the world of parasitology. So I started out studying toxoplasma and plasmodium and studying those diseases. And I think it was really there that exposed me to this world of kind of host pathogen interactions. I'd always been interested naturally in cell biology because that was fascinating, but then kind of recognizing there was a whole nother level to this world, when you have infections and that, there's all this interplay and crosstalk and defense and evasion between hosts and pathogens. That's really what I think took my interest in science to the next level. And again, just kind of made me feel really comfortable that I could do this forever. Absolutely. This is so interesting. And so as far as kind of deciding to pursue a PhD, that's where that came from. And so I was really interested in parasitology. So I focused my applications and basically the best programs I could find, and really was hoping to get into some of them, and ended up being lucky enough to get into Johns Hopkins, where there's a huge malaria research core. And that's what drew me to the university. But it was actually kind of some more twists and turns to get me to where I'm at now, and obviously in the field of virology. So after getting started and doing rotations within the program, I decided, what, why not switch it up again? Let's make things interesting and get exposed to a new field. And so I kind of took a left turn and joined a mitochondrial biology lab that was studying mitochondria in the context of a lot of different diseases, both infectious and non-infectious. And I did my thesis research in that lab for a year. And after a year, the PI of that lab ended up deciding to move their lab to Germany. And that was a little bit of a further move than I was kind of wanting to make. So started to look for other labs in the department where I could find a home. And it was kind of a natural transition into Andy's lab because there was a collaboration that already existed looking at mitochondria in the context of viral infections. And so I really kind of sunk my efforts into that project, that collaboration, joined Andy's lab, and have been, so I'm definitely relatively new to the field of virology. And so that's intimidating, but also super exciting because there's so much, that wave of learning and new experiences and new people and perspectives is, it's always such a fun time. So that's kind of what landed me here. I kind of started out parasitology, then more cell biology, and now I'm, kind of back to the world of pathogen interactions. So.
Right. And can you tell us a little bit about your lab and then maybe talk about sort of your thesis work, its sort of the bigger goals, and maybe some of the techniques that you use to do it?
Absolutely. So Andy's lab is a super fun lab that's kind of before the pandemic would say primarily interested in influenza, characterizing influenza viruses year to year, kind of the differences that help them replicate or inhibit their replication or escape of immune responses and things like that. But the pandemic hit, and then there was a huge branch of COVID research that sprung up in the lab and is still going. So we're at the moment kind of 50-50 split between influenza and COVID. And my research is kind of a little bit of a unique niche within the lab in that the lab is interested in a lot of different ideas, but the one that's kind of most centrally focused of my research is the idea of this temperature gradient within the respiratory tract. So the upper respiratory tract in the nasal cavity is a cooler microenvironment at about 33 degrees Celsius than the lower respiratory tract, was closer to that core body temperature of 37. And so the lab has done some work in the past to characterize how these changes in the microenvironment of these temperatures influences viral replication and host immune responses. But there hasn't been a whole lot of work done characterizing the changes in host cell biology at these two temperatures. And so that's where kind of my project steps in. So my research focuses on characterizing changes in mitochondrial biology at the two temperatures of the respiratory tract. And, after kind of establishing that characterization, we're interested in kind of relating that to how these changes in host cell biology really influence and kind of are directly connected to the changes that we do see in viral replication and host immune responses.
And I guess to just ask more broadly, are these differences in temperature, do they have effects on sort of the pathogenesis or the ability of viruses to infect different cells?
Yeah, so it's kind of one of those answers where it's applicable in so many different settings, right? But so we're looking at kind of a couple of main things right off the bat. So the changes in temperature, definitely we do see, and we know from prior experiments that there is changes in kind of the replication rate. So if we're looking hour to hour during the course of infection, what's differences between those temperatures. So we do see differences there. And we also see differences in kind of peak titers that are reached between these environments. So that is what starts to make it pretty complicated, as far as, does it matter if it replicates more slowly, if it reaches a similar titer, or, go down that rabbit hole. But we kind of see differences across the board. And one of the things that also adds a layer of complexity to it is that different viruses display different differences. So year to year and strain to strain, it can vary as far as, kind of those differences that we see.
Right. And I mean, I know for like SARS-CoV-2, there's a lot of differences just between sort of the ability of the virus to infect the upper respiratory tract, the lower respiratory tract. And some of this has been tied to, the binding affinity maybe for the receptor, but it makes you wonder if there's some other factors, like this that are also involved as well.
Yeah, and that's where, so we kind of try to pull in as many different techniques as we can. So there's, kind of some of the things we're interested in doing are metabolic profiling, and that kind of gets at the host cell. Are there's differences like fundamental metabolic profiles of these cells that influences their ability to mount an immune response, and maybe that is what explains some of the differences. And but mixing in other techniques like RNA-seq to see if there's differences in the translation profiles, and all of these kind of different ways of looking at it. Is it host cell mediated? Is it some just maybe environment mediated? And seeing whether or not there's any kind of, at least across these different settings, because like we say, there is differences that we see between viruses and things like this, but is there something that kind of holds throughout all of these different settings that we can kind of point to? And I think one of the most exciting settings to think about this is kind of for vaccine purposes, and kind of how can we maybe tailor a vaccine to really boost an immune response in a certain area or make up for a deficit in another. So, hopefully if that's the end goal, then we can, end up recognizing differences between the upper and lower respiratory tract and being able to target them for therapeutic purposes.
And are the experiments that you do, are you using sort of cell lines, or is this in vivo work? How do you study it?
Yeah, that really gets to kind of your point then too about tropism and things like that. So we use both immortal cell lines and primary human nasal epithelial cell cultures. And so some of the kind of initial experiments we've done as far as temperature related, it's great and kind of much more high throughput to do the immortalized cell lines, get these initial characterizations. But it's really amazing that we have the ability to go then to these primary cell lines that really represent the nasal epithelium to get at questions of, okay, are we affecting different cell types in different ways? Are we seeing differences in infection based off of one cell type generating the main phenotype we're seeing and things like that? So I think that's one of the kind of the most interesting aspects of the research in that sense too, because then you can get as close to your basically in vitro what you can get to what we're seeing in our nasal passages.
Right. And I guess, I was reading recently, there's like new liquid air interfaces, different systems in a way that are even fancier, more physiologic, as it were. So you can get sort of more and more closer to the in vivo host, as it were.
Yeah. So our culture systems do use that kind of air liquid interface. So we differentiate them into kind of the major cell types that you do see. So we have ciliated cells that are, vibrating with their cilia on top, and their cultures are super interesting to look at. But like you say, this kind of technology is advancing so fast. You see these, larger and larger organoids that represent these things. And it's so fascinating to see the advances that are kind of being created. And that's what I think is really cool, because as far as, again, the kind of for the field of virology and host pathogen interactions, I think you are able to merge so many different things, because you have these representative cell cultures that are combined with, I feel like a lot of times it's kind of these almost physics-based labs that are building the organoids. And then my previous lab did a lot of imaging. So kind of taking these advanced imaging techniques to these cultures, and combining, obviously the omics world is large. So combining all this together in this setting is, I think, super exciting.
And I guess, are you presenting some of this work at ASV this year?
Yeah, so I'll be presenting a poster kind of highlighting the initial characterization that we've done, the differences in the mitochondrial networks between the upper and lower respiratory tract. So I think that the results I'm excited by, we see some interesting things, and that there is differences in the mitochondrial networks. There's differences in membrane potential, some functional characteristics of the mitochondria. And kind of, as I mentioned before, we're really excited now to kind of take this to the infection setting and tie it back to the immune differences in immune responses that we're seeing. So I'll be presenting a poster in the innate immune group, and and it's one of the, like I said, this is quite new to the field still. So this is something that I'm super excited about to kind of obviously get the opportunity to talk to people, see what they make of the research, the findings, the initial characterization, because getting into a field, there's a lot that you try and learn, but there's always so much that you don't know. So I think something I'm super excited about from a learning perspective.
Cool. And could you talk, so, some people may know about the mitochondria, and sort of how that interacts or interfaces with immunity, but can you just give us a brief summary about kind of what is known about that? What is the mitochondria? How is it involved in innate immunity?
Yeah, so mitochondria, I think, are super interesting organelles because they're so important during homeostasis for maintaining cellular health, but also during infection settings. So especially when we're thinking about the respiratory tract, we're thinking about a lot of ciliated cells, pushing the mucociliary escalator, forming those tight junctions. All of these kind of functions of the respiratory tract in a lot of ways are mediated by mitochondria. So the movement of the cilia needs a lot of energy to occur. So mitochondria directly provide the ATP necessary for that physical barrier to infection. Mitochondria are calcium stores. They're generators of reactive oxygen species, and kind of that ion and nutrient homeostasis is really important for, again, cell health and those tight junctions and that physical barrier in the respiratory tract. But during infection, they're even more important, you could say, because they're kind of central mediators of innate immune signaling. So kind of in early stages, they're signaling platforms for MAVs that kind of facilitate the induction of the type 1 interferon response, and that's kind of the general antiviral profile. And in maybe more severe settings, when the cell has made the quote unquote decision to undergo apoptosis, the permeabilization of the outer mitochondrial membrane and the release of cytochrome C is important for that induction of apoptosis to kill the cell, which presumably limits viral replication. So both during, kind of the maintaining the physical barrier to infection and mounting that innate immune response, mitochondria are incredibly important organelles. And there's more work that is a little bit outside the scope of my project, but a lot of groups are starting to look at, metabolic profiles of adaptive immune response T cells and B cells, and how the metabolic profile of those cells really contributes to their effector function. So definitely important organelles, in something, I'm always going to be on their side. So that's my fight for them.
Are there enough differences in the way that mitochondria function in different species that they could also be a determinant of sort of permissivity or restriction?
That's a great kind of topic to bring up. I haven't thought, and you mentioned this earlier as well with the avian flu and kind of thinking about maybe crossover events or things like this. It's actually not something that I personally have thought a lot about in my project, and this is exactly what I was excited about for ASV, getting to interact with virologists and get their perspectives. That's super fascinating. I don't know a whole lot about differences in mitochondrial networks between humans and animals and things like that, but it's absolutely something I think would be super interesting to look into. And
yeah, like you wonder how conserved like the components are there sort of species specific components, things like that. It's obviously a very core function.
Yeah, so a big topic of my previous lab's research was looking at mitochondrial quality control. So, every now and then mitochondria become damaged, or proteins kind of become damaged, and you need to either or get rid of whole mitochondria or mitochondrial proteins. And we've learned a lot about how human cells do this by studying yeast. And so, there's a lot of overlap, but it's not identical. The proteins and the pathways are not identical. So, kind of based off of that, I would assume that there's definitely some overlap, but surely some unique differences as well. And be really interesting to see if those differences come up in areas that are important for antiviral signaling.
Right. So I guess then just to finish, it sounds like you're fairly early in your PhD. Do you have any sort of early reflections about where you want to go after your PhD? Are you interested in academia or industry? Do you have any sort of notions about that?
Yeah, so it's something that I've kind of been exploring. Our department has a lot of great resources. So I'm kind of surveying the landscape now. And I think that there's a lot of what I guess the thing that excites me the most is that there's a lot of opportunity. And so I'll have to kind of spend some more time over the next couple of years deciding which exactly avenue is going to be kind of the best fit for me and my personality. But I mean, I think the one thing that the pandemic has kind of shown is that this research, and that's another thing that excites me about virology, it's not happening in a vacuum. And there's a lot of importance to the basic research that gets conducted a lot of times at right, universities and things like that. But there's also the potential to hugely impact the world of public health through the creation of vaccines and things like that. So I've always been focused on academia because I think that academia does a great job of kind of getting you ready for that environment. And I think that there's some extremely unique things about that. So I'm really happy to be a part of an academic setting right now. But that's one thing I do think the pandemic has kind of shown is that there's a huge possibility for impact moving into a government space or an industry space where you're able to kind of do more of the translational side of things.
Right. And then just to finish, I guess, what has the last 2 years been like for you? So it's not an easy time to be a graduate student. It's, during the middle of a pandemic. So how's it been like for you sort of personally and professionally?
One thing I'm super thankful for is that I was able to get into my program kind of the year before the pandemic hit. So I was able to get that first year of in-person building relationships and connections and learning the environment in person. So that's one thing that I think that was really nice, kind of grounding me throughout shutdown and virtual work and everything like that. But I think it's kind of weird because I think, at least, I don't know if this is just my perspective, but I think a lot of my personal efforts and talking to my kind of fellow cohort mates is that we all spent a lot of time trying to push things forward as if there wasn't a pandemic, trying to maintain progress and kind of find a way to make it normal. That I don't know if there's been a whole lot of time to kind of reflect and see where those changes have come up. So I think a lot of effort has just been kind of fighting to maintain what was before. But I think that one thing I'm noticing now definitely is how excited I am to go back to kind of in-person large events where you're really interacting with your department, or obviously things like conferences where there's a whole lot of new conversations and new ideas floating around and things like that. So I think that as far as kind of a detrimental impact, I think that kind of loss of conversation has been something that I'll miss, or at least kind of retrospectively will miss. But at the same time, I think it kind of mentioned maybe it's almost something lucky to experience as a grad student in the field of virology, in the world of virology, because it's amazing. I think one of the biggest maybe complaints or shortcomings of like the academic setting is you do spend a lot of time at the bench, and doing that basic science, and it's so important, and that's why we're here. But it's really amazing to get the opportunity to see that it really is it's so important, and getting to see the coronavirus researchers kind of, that we're doing this in the dark when nobody was, quote unquote, right. So it's obviously there's lots of great research that is appreciated, but that kind of comes to the forefront, and all of a sudden CNN wants interviews with them, or New York Times wants to interview them for articles. It's really amazing to see that the things that we tell ourselves that make this research important and make this work important really are true. And so I think that's something I'm thankful for, having kind of looking back at the process and everything.
Great. All right. Well, thanks so much. And we look forward to hearing about your work at ASV.
Really appreciate you having me. Thank you very much.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.